With that, it's my pleasure to introduce Dr. Sugana Severaj, who is founder at Kirchsteig, bioresearch consultant and mentor, and also organizer and moderator of today's panel. She will introduce the topic of the panel and then our panelists. The stage is yours. I think you're unmuted.
Yeah, thanks, Monika, for the introduction. I have a great pleasure in introducing the esteemed panel speakers. We have Dr. Nitha Radhakrishnan, Additional Professor and Head Department of Pediatric Hematology, Oncology, and BMT, Postgraduate Institute of Child Health, Noida. She holds several prestigious positions. She's the chair, Childhood Cancer Working Group, past secretary of International Pediatric Association, Pediatric Hematology Oncology Chapter of Indian Academy of Pediatrics. Her research interest is on improving childhood cancer outcomes in resource-constrained settings. And she has published more than 70 peer-reviewed papers. We welcome you, Dr. Nitha Radhakrishnan. And next on our panel is Dr. Vaishnavi Sambandam. She has played key roles in both academia and industry in cancer domain. She has a PhD in cancer biology from Temple University. She has worked on hepatocellular carcinoma, and she has done her postdoctoral fellowship at MD Anderson Cancer Center, where she studied head and neck cancers. She also investigated tumorigenesis pathway in lung and ovarian cancers in Mayo Clinic. And she has worked as a scientific team lead, scientific liaison at Champions Oncology. And she has spearheaded development of ex vivo models, immuno-oncological assays. Currently, she is working in NCCN as an oncology scientist. And next, we have Dr. Sushma Patmaja. She earned her PhD from Center for Biotechnology at Anna University and postdoctoral studies at Rajiv Gandhi Center for Biotechnology. With 14 years of experience in genomics, clinical diagnostics, and multidisciplinary research projects, currently she is the head clinical genomics and chief scientific officer at Kaiwa Genomics Inc. She heads the development of diagnostic products in the field of oncogenomics, pharmacogenomics, and nutrigenomics. As an expert in NGS data analysis and clinical interpretation of variants, she's translating complex genomic insights into actionable healthcare solutions. And she's committed towards advancing personalized medicine. And we welcome all the panel speakers for the event. And let me introduce the topic as such. And we all know there is a heavy cancer burden and it is growing. And this is a report from WHO, Global Observatory. And there is a predicted 77% increase in 2050 from the estimated 20 million cases in 2022 to a predicted rise of about 35.3 million. This is worldwide scenario. And also in India, if you see, there is also a great rise from 1.41 million to a predicted 2.69 million in 2050. Next slide, please. And there are a lot of challenges in cancer treatment involved. Cancer treatments are very complex and intense, and they're very expensive, toxic, and protracted. And as such, there are other problems like cancer recurrence, late complication, and there is lifelong surveillance is involved. And the outcomes are also unknown. And the availability and affordability of new therapies are also in question, actually. Next slide, please. And there is a need for patient-centric care. Though we have done a lot of huge strides have been made towards understanding the cellular and molecular progresses underlying tumor progression, this has not been completely transformed into patient care. So there is still a struggle to find a cure for cancer. And the current therapies have their own limitations, and there is increased survival rates. Though it has increased survival rates, but the standard of living is compromised. So there is a need for patient-centric care and research. Next slide, please. And there is WHO has recently emphasized to move in from tumor-centered model to a patient-centric model, wherein the patient is the center of the thing, where all the stakeholders they try to move the journey of the patient smoothly. And this is a multidisciplinary aspect wherein clinic as well as researchers and other support staffs work together to support the patients. Yeah, next slide. Next slide, please. And so now we have the webinar. Given these gaps, we have eminent speakers in the panel who are there to address the embracing patient-centric cancer care, and the mainly will be addressing the importance of radical collaboration within clinics, within healthcare providers, academia, and industry partners. And we'll be discussing lot of interesting questions, like the panelists will be addressing a lot of things like what is patient-centric care, why it is needed, what are the key gaps in it, and what research is needed, what are the challenges and barriers, and how can we translate this lot of newly acquired knowledge is there. How can we translate the newly acquired scientific as well as clinical knowledge into patient-centric clinical improvements? So next, we are done with the intro for the session. And now we have questions for the panelists. And I welcome all three panelists.
Yes, thank you, Sukanya, for the introduction. And it was a good introduction by you.
Yeah. Sukanya. So we are moving into the questions for the panelists. And the first question is for Dr. Vaishnavi. Can you throw light on what is patient-centric cancer care and what is the need for patient-centric cancer research and cancer care?
Yeah, thank you, Suganya, for the nice introduction. And it's nice to meet the fellow panelists, Dr. Neeta Radhakrishnan and Dr. Sushma Padmaja. And thank you, GBR, for this opportunity to participate in this important discussion. So to answer your question, Suganya, the picture that you showed in the previous slide of how you put patient in the center of the different cogs in the wheel, and that's how we have to approach patient centricity, right? And the concept of patient centricity has emerged in the recent past, like about 2 to 3 decades, where we started talking about patient centricity. And there is no real definition of what patient centricity is, but in simpler terms, it's where you put the patient's need in the center and you systematically engage the patient in different steps in the drug development process and in the decision-making process during treatment. So we need to cater to the patient's need. And where in cancer research, when you think about it, how can you put patient centricity in the center is understanding the disease setting and also trying to understand and identify the unmet clinical needs, which will eventually direct your research questions and how you approach your research hypothesis. So that is where patient centricity comes into picture, right? And when you think about cancer research, whether it can be academia or industry or in the clinical space, collaboration and communication are the key aspects of having an engaged patient-centric approach. Gone are the days where the academic research was siloed and it was standalone in a small lab in a university, and the end product of the research has never seen the light of the day and it has never reached the patient. But now we take a very multidisciplinary approach where we need to constantly engage all the different aspects of the research and the entire cancer care continuum from the beginning to the end, so that it actually reaches the patient. And that's how I see patient centricity approach, especially in the cancer research aspect of it. And I would like to hear others' opinions as well.
Yeah, that's a wonderful explanation, Vaishnavi. And the next question is for Dr. Neeta. Can you explain how you offer patient-centric cancer care as an oncologist? How do you do this?
Yeah, thank you, Sukanya, and thanks you to Global Biotech Revolution for this discussion today. So, and the topic you have selected, and congratulations to you for the topic that you have selected. It is something very new and something which has evolved over the last few years, and it's something which we are practicing in our daily lives. So, a lot of years ago, when I went for a meeting, I was pleasantly surprised when I started seeing. In chronic diseases, patient-centric approach has been there for a long time. Cancer care, incorporated recently, but there are diseases where you are in a conference, you are sitting with the patient and the doctors in the same room and listening to the same talks. So that was a concept which was very alien to us here in India. But over the years, we have seen that happen across diseases, and now this is something which we are incorporating in our care also. So whenever there is a patient who is diagnosed with a malignancy, and when we offer treatment and when we give them the options that are available for treatment, a lot of emphasis is given to family's opinion. We want to know what the family wants to achieve from the at the end of the day, whether the family is willing to stay back and get the treatment done. For example, there are malignancies where saving the life would require amputation or removal of an eye or certain organs which are very very crucial to save the eye. But at times there will be complete no from the family, and then it's like for their social environment where they live in, having a child with a disability may be more difficult than having a child with as a cancer survivor. Yes? So these are issues which we face on day-to-day life, and a lot of discussion happens before we finally adhere to that, and we decide on the path to be followed. And second, whatever treatment is available globally, there are limitations to what we can offer our patients. So many a times this may not be to get the treatment available in locally, that might be difficult. The quality of treatment may not be as good as it is in high income economies. Then it may not be what the patient also wants. So there are a good percentage of patients who still opt for complementary medicine, thinking that there are no side effects, and so these things keep coming up in our practice regularly. And so putting the patient at the middle or the family at the middle. When I say I keep saying family because when I look after children only. So for me, a child with the disease means there is a whole family around it whom we have to target, whom we have to counsel, whom we have to get into confidence to start treatment. So putting them at the center of it all has been helpful, and that is how we make the decisions in day-to-day practice.
Yeah, nice to hear from you from the clinical side how you apply patient-centric care. Thank you, Nita. And Dr. Sushma, as a person from industry, how Kaivar Genomics, your company, is involved in patient-centric cancer care and in specific on personalized medicine?
Okay. Hi, Suganya, and a very warm welcome to all the rest of the panelists and who are listening to the webinar. So let me just introduce what basically Kaivor is up to. So Kaivor is a personalized genomic company which was started way back in 2013. So this was the first company in India to basically concentrate only on cancer genomics. And I would just like to elaborate on two different products that actually, caters to the personalized medication space. One is our product, which is called as a Canalytics, which looks at the whole exome of a patient to calculate the complete TMB, which is called as a tumor mutation burden. Basically, by knowing the tumor mutation burden, you can say whether a patient is eligible for immunotherapy or not. And apart from that, this test also gives additional information of what are other targetable mutations present in that particular tumor sample. So it will have actionable mutations, which will be targetable. Then you'll also have prognostic diagnostic markers, which are given along with the report. And our second product is a Medlytics product, which we call as a pharmacogenomics product. This basically this analyzes a person's DNA to understand what is the capacity of a person to metabolize a particular enzyme. So basically, we look at most of the drug metabolizing enzymes. It is also looking at whether a patient will be suitable for a particular drug and what will be the dosage that is recommended. So there are global efforts which have gone into formulating a lot of prescribing methods to actually identify what can be given for a particular metabolizer type. So we do this Medlytic test, and it covers almost 200 plus medications which are already present present, and FDA also kind of gives labeling for these particular biomarkers. So these are the two products which physicians and patients can use to identify what kind of targeted treatment or what kind of adverse events can be actually avoided by taking these tests.
Yeah, thank you, Sushma. Thanks for throwing light on what Kaiwa Genomics is working on. Yeah, Dr. Nita, as an oncologist, what do you want to communicate to the research community as if now most of the participants are from research. Like, what do you want to tell for the research community? Like, what is happening in your clinical setting? Like, what are all the needs of cancer patients? What the advanced stage patients? What is the requirement for the advanced stage patients? And what type of future therapies and diagnostics are needed? Dr. Nita, can you throw light on it?
Yeah. So research is a very integral part of our lives. And even as clinical clinicians, we do engage in research which improves the quality of the treatment that we are providing to patients. But definitely, bench research is something we are not able to dedicate as much time for. And for all the researchers who are out there who are working on cancer treatment, the requirements from the clinical community would be three. One, we would require more and more targeted treatment because gone are the days when you treat when you flat a swine with a machine gun. For a fly you need to have a fly swatter, you need to have a particular treatment for that particular malignancy. You cannot keep bombarding all the cells in the body, all the rapidly growing cells in the body with high doses of treatment which used to happen in the past. And so in the past we would see patients, of course, having a lot of toxicity of treatment. And we also would see a lot of late long-term side effects also. So in the earlier days, patients who would have received like radiotherapy, a lot of radiation to the brain, or a lot of chemotherapy, which was in those days, it was approved as treatment. We would see long-term side effects in those children now. But nowadays, we try to titrate our treatment in such a way that the toxicity comes down with every passing year. With every passing new cooperative protocol, there is a whole emphasis on de-escalating treatment as much as possible, avoiding radiation whenever possible, avoiding high dose chemotherapy if that is an option, and risk stratify the disease so that you optimize the treatment. You do not give the entire treatment to all of them. Only the patients who actually receive require high risk are receiving high risk. The rest of them maybe you can get away with lesser and lesser treatment as you go on. So the one big advantage and the one big advent in this is that with targeted therapy, like was mentioned earlier. So nowadays we have so many actionable molecular mutations that are available where targeted treatment can avoid or give away with chemotherapy completely. So that is one advantage for us, that if we have more and more targeted treatment for our cancers, then definitely that will help us in the long run. The second request to the researchers would be to ensure the quality of the product. So there are products which come out by multiple companies would bring it out, but there has to be ongoing research to look at the quality, the surveillance of that the product which has come out, and how that drug has actually benefited or not. So there should be an ongoing work on that. And finally, with the same drug, we should be able to find out. So that's something that is again clinical research, that's not bench research, to find out how better protocols or protocols which have been modified to the situation, so that they're called adapted protocols. So the same protocol in US may not be used here because our scenario, local scenario, infective milieu, etc. is different. The child's nutritional status is different. So we adapt treatment protocols to our local environment with our available resources, the patient's available resource. If a patient is living 300 kilometers away, then you would adapt the protocol accordingly so that we are able to deliver treatment with the minimal toxicity. So that is again an area of big research. So these are my three big asks for the research community.
Thank you, Nita. Thank you. Throwing on what need for targeted therapy. More new drugs are needed, but as well it is some research on the existing drugs to adapt for the condition for the patient. particularly for the patient, is also needed. Yeah, that's great, Nita, to hear from you. Dr. Vaishnavi, can you address how this type of communication, like from the oncologist, can enhance the research output? Why this communication is important? And in general, what are the takeaways as scientists and researchers can have from oncologists, and vice versa? Like, what can oncologists gain from researchers, and why this communication is important?
Yeah. So before I answer that question, Dr. Nita, it was really great to hear about the importance of patients in the clinical research, at least in the way how you treat the patients, right? So I work at NCCN, the National Comprehensive Cancer Network, and we do have treatment guidelines on which we actively engage a big panel of experts from different clinical hospitals, and we do help in developing and updating clinical content every year. And in that panel, we do have a patient advocate, and they do talk about what needs to be addressed. Like when we talk about like different therapies, they do mention about the side effects and address all those issues. So patient advocacy has become a key component in the entire cancer care, which is really nice. And when you talked about high resource, high income resource, mid income, and low income resources, and we do actively we are involved in developing those frameworks, which will set in like basic resource and core and enhanced resources, which can be adapted to low, mid, and high income resource countries. So I'd like to talk to you offline about it, and we're always looking for international reviewers who can review these contents to make sure that it is still relevant to those countries and whatever is available locally. And whatever we talk about here might not be relevant in different countries. So that was really a great insight that I took from you in today's talk. And to answer your question, Suganya, sharing the perspective of clinicians on research is really important for us to have a good research hypothesis and to have a good research project in general. So from my experience, I can talk about my translation work at MD Anderson, where even at the beginning, when we had a research objective, even to frame that objective, we had a team, a multidisciplinary team, which involved different clinicians and bioinformaticians and other fellow peer academic basic researchers to make sure that the project has a good goal, which is again has a good patient relevance, which can then be translated into the clinic. And that was so that's how we developed the hypothesis at the beginning, right? So it's not just to answer one question, okay, what does this gene do, and how can I? So how does this protein work? So those are questions which are again which are important that needs to be addressed. But how can you make it relevant to the patients, right? So I was working on um, and identifying biomarkers of sensitivity to PI3K pathway inhibitors. So even from the beginning, we wanted to be sure that this, at the end of the research, whatever comes out of it, it has to reach the patient. And that's how we had the research objective defined, and that's how we had the framework done. So at the end of the research, I was not only able to publish the research, but also I was able to take it to the clinic, and we were able to initiate a phase 1 clinical trial. So I was able to see the fruit, to reap the benefit of the work that you do, right? And not everyone has that opportunity, because at the end of it, once you're done with your project and when you're looking to write your manuscript, that's when you think about patient relevance. And that's one paragraph at the end of your publication. And that is not the time when you have to be thinking about patient relevance. That has to be at the beginning of the research project. And then it also helps to have frequent meetings with these clinicians so that they also keep you on focus. So you can have as many research questions, like a lot of questions out there, which can digress into different directions, and you lose focus on the patient centricity aspect of it. So having meetings with the clinicians also helps you to keep a track of your progress and be focused on patient care as well. So that's how I see where the clinicians can be part of the process in having a strong research objective. And the other aspect is the perks and incentives that the clinicians can get out of it, out of a collaboration, right? So co-authoring publications is one, and even having these clinical trials, like again, co-PIing, like different clinical trials. So what does the clinicians get out of it? That is also something that we need to pay attention to. Of course, the researchers get a lot of insights and inputs from the clinicians, but what are they getting at the end? So having those things sorted out will really help in strengthening these collaborations.
Yeah, nice to hear your own experience about how you coordinated from the research to the clinic. Yeah, that's nice to hear. And
Thank you.
very happy to see like a collaboration happening here itself, like you want to collaborate with Dr. Nita. That's a wonderful thing. Yeah. Yeah, thanks, Vaishnavi. And now moving on to Sushma, Dr. Sushma. Uh, like we heard from clinic and academic research and things like that. Dr. Sushma, can you tell from the industry perspective how this type of collaboration is happening? Do you actually collaborate with the clinic? Do you interact with the clinicians? Yeah, very happy to hear from you. Yeah. I think you're muted, Sushma.
Yes, yes. So what I would like to throw light on from the perspective of how, this gap is being filled. I think Dr. Neeta had addressed that, there is a requirement to address more targeted therapies and how, but what we are presently doing is how to, take whatever is already existing to, employ in clinical practice. So basically, we work closely with oncologists, and not only oncologists, we also work for the adverse drug reactions with various other doctors, basically, also cardiovascular and psychiatric medications. A lot of other doctors do come for, adverse events and everything. To be very specific about, cancer therapy and, oncology treatment, this has been grown tremendously from the past where, like, Vaishnavi has been part of NCCN. So NCCN has given these right recommendations. So it is very difficult for a doctor to keep updating on all these things. So as an industry person, we as molecular pathologists, and we are there in the molecular tumor board, where we give insights and updates from all these regulatory bodies, which is very important in clinical practice. So basically, doctors, yes, they are very well aware, but it is a very this field is growing immensely large. So what happens is there are a lot of research that happens and a lot of publications. So as and when we are looking into the molecular phenotypes, so basically we do a full exome sequencing. So what happens is uh, uh, in the industry before, it was single gene sequencing, where you're targeting just a single gene and seeing whether a particular targeted therapy is applicable. So nowadays it's been the CGPs, which is comprehensive genomic profiling, where you look at least 500 plus genes to see what are the different molecular alterations. So what we come across is these targeted therapies, when actually come in, as a co-mutation, or they exist as a molecular profile, they have some discrepancies, and there are a lot of differences. So each patient is very different. So what oncologists feel is that though there are a lot of large clinical trials which enroll large number of patient cohorts to understand what is the therapy, then also they have a lot of other doubts, as in when it comes to co-mutation, coexisting molecular profiles, very different perspectives from different patients, where some patient will respond with the same particular biomarker, but another patient will not be responding. So these are the differences, and we have been part of molecular tumor boards. We have been part of a lot of molecular tumor boards, wherein the doctors present their cases, and they also present the genomic reports to us. And whatever genomic reports we present to the doctor, we make sure that we have a one-on-one discussion with the doctor. And our report is also an extensive report which characterizes the molecular phenotype of of a patient. So it will tell you what are the targeted drugs that are available. It will also talk about clinical and case studies, which we keep updating, especially when there are co-mutations, when there are, larger cohort studies, very important studies from, different papers like, ASCO, ESMO, and all these recommendations, whatever we get on an updated basis, we make sure we let the oncologist know what is the, treatment outcome. And I think it's a very great task for an oncologist to treat patients because they have a number of patients. So the care gap in understanding, the molecular treatment, molecular profile, getting in that targeted therapy and making it useful for the patient who's taking it, especially for him if he has targeted, obviously there's not going to be a necessity to involve him in a chemotherapy regime. Or if you also want to treat with a chemotherapy, we can also look into what are the drug adverse events. So we can look into the pharmacogenomics. So these are the care gaps which we are trying to fill. So researchers do have worked on this, and we have bodies like NCCN and FDA who are giving approvals to biomarkers. So we play this gap filling of taking this technology and these therapies to the oncologists so that they can rightfully use it for their patients.
Yeah, great to hear. Like, can understand how extensively as an industry you are actually working with the oncologist and you are assisting them for all these molecular diagnostic works. Yeah, and thank you so much, Sushma. And next question is for Dr. Vaishnavi. What perceptual changes are occurred in cancer research? Can you throw light on that? Like how the research should go on? What perceptual changes are occurred?
Yeah, so when you think about research and how you put patient at the center of the research, you think about it as a series of concentric circles, and then at the end of it is at the middle is the patient-centric approach, and then you have all these layers of in vitro research, in vivo, and then you do clinical research, and so on and so forth. I think the perception change here would be thinking of it as a Venn diagram of three intersecting circles, right? And those will be the industry research and academic research and the clinicians. So, and the center of those three intersections will be the patient at the middle. So not just all of these circles should focus on the patient at the middle. At the same time, those two circles also need to have the intersection strengthened, right? The industry and academic collaboration that needs to be strengthened, and the industry and the clinician space that needs to be strengthened. Of course, highly regulated, and the industry and the academia and the clinical space that needs to be strengthened. And again, we need to, like I said, we need to think about the perks and incentives and how those environment can be safely regulated and at the same time be strengthened for us to achieve the common goal of patient relevance. So any kind of R&D work, it starts in academia and starts in an academic lab. You have this basic research question framed, and you have a set of outputs which then gets carried over to industry, and that's where the clinical trials happen, where from preclinical space it takes it to the market. And once it is in the market or once it is in phase 3, then that's when the clinicians start prescribing it to the patients, right? So it is a continuum, and at every continuum, we need to make sure that all the stakeholders are actively engaged from the beginning to the end. And that is a perceptual change that is needed right now. So not any one party cannot be siloed and one party cannot just work on themselves. You need to have a multidisciplinary approach. You need to think of different dimensions of research. And again, when we talk about preclinical research, doing in vitro research, like you do it in cells, and then you get the output, and then now you move on. Then you move on to in vivo and you work on animals, right? So, but then those models do not really address the toxicity issue, for example. In vitro, it's just tumor centric, and you make sure you kill the tumor cells. When you take it to in vivo, it still doesn't reflect the human toxicity because of physiological differences. And the field is also evolving and it is updating itself to address all these issues in the patient-centric world, where now we talk about the microfluidics approach and organ on chip. When you talk about those newer technologies, where it's no more in vitro or in vivo, again, the progress being ex vivo. Okay, you can take the tumor outside and you can make organoids out of it and try to make sure that they are getting killed and your drugs work well in an organoid space. Now you have the tumor microenvironment that you can build in to make sure that everything works together and you get the output that you need. And then comes the organ on chip, especially when you talk about tumor on chips and the combining organ on chips. Now that is a space where it reflects the patient, it reflects the human model. So now you can actually, in real time, study the drug toxicity effect in a smaller microfluidics chip. So those are technologies that are being upgraded and updated, keeping the patient at the middle. So as cancer researchers, we also need to update ourselves in the newer technologies to make sure that it is still catering the needs of the patients and not just stick with the in vitro model. Again, it depends on the resources and the constraints within the field, but to have an open mind and understanding where the field is going, that really helps building better research and helping to strengthen the cancer care. Continue.
Yeah, thanks for your broad vision and perspectives about like what changes are occurred in the cancer research. Thanks, Vaishnavi. Dr. Nita, can you explain the challenges in treatment? You have said about the side effects, and can you throw more light on drug non-compliance issues, side effects, drug resistance, and treating terminally ill cancer patients? How do you go about that?
Okay. So whenever we start treatment, it's usually treatment is with a curative intent. Especially in a pediatric population, whenever we are treating, we are usually for most of the pediatric treatment cancers, unless it's an extremely advanced disease and it's beyond any treatment options, that is a very, very small percentage of patients. For majority of other patients, it's usually treatment with a curative intent. So you have a cure at the end of it, that is you're aiming at, and at the end, after completion of treatment and another 5 years from diagnosis, if we go by, then you label them as survivors. So that's what everybody wants: that treating the patient, the treating the family, as well as the treating doctor and the medical fraternity in the team. So for whenever we treat with that kind of an intent, of course, the challenges that we would face is, one would be that we need to ensure that we get the best treatment option for the child. We would adapt the treatment to the child's, like I said again earlier, that it would be to the child's nutritional status, as well as the child's any additional toxicity. If the child has some the additional unusual toxicity or cancer predisposition, anything else, that of course you titrate your treatment according to that. You would want to give the best quality of drugs. So that is again something which has been heavily debated upon or heavily discussed in the last few years, that there are, with the advent of so many new players in the market, there are so many drugs which are out there which require a very good quality control. So this, as an end user, we are always worried about what product we are using and whether this will actually, if we use it, which will actually give good benefit to our patients. And finally, we want to prevent what is completely undesirable, which is a treatment related toxicity. So it could be a toxicity in terms of side effects of the treatment that has been received. It could be infections that are part of, like, infection is again a toxicity because you are creating immunocompromised patient, as in a patient who's immune compromised because of the cancer per se. On top of that, there is immune compromise because of the drugs or the treatments that we are offering, whether it is chemotherapy, radiotherapy, bone marrow transplantation. All of this would have complications in terms of toxicity. So that's something which we want to completely avoid, and definitely prevent any mortality related to that. So as a whenever we treat with a curative intent, this is what we aim for. So the risk adapted treatment with the best drugs possible and the best efforts so that we reach cure. So that is when we do it the first go. So whenever we do treatment for terminally ill children, like you discussed, so terminally ill children means there are treatment which is beyond the scope, there the whatever we when if despite whatever we offer, there is still a big chance that there would not be a cure. So then this is again discussed with the family, and we give them a very, very realistic counseling as to what is to be expected, what is the effort, what is the struggle ahead, and what are we expecting. So if we think that it is beyond the percentage or the chances as per the current data, current evidence that we have is that the child may not survive. So it is like a second relapse, or if it's a very high risk disease with a bleed in the brain. So these kind of scenarios we do encounter once in a while. When we sit down with the families and discuss what are there, that then we revise the treatment goals accordingly. So our treatment now is to provide comfort care in that scenario. So you give them, again, there is a big role of all medications there as well. So there is a lot of medication involved in pain relief, in symptom relief for these patients. There would be palliative chemotherapy or radiotherapy options where that. So you give treatment to keep the disease under control, so that the family or the person, the family has the child around or the patient around for a longer period of time. So for adult patients especially, it's a they always want time so that they can, they can say goodbye. So that's a very, very crucial part of treatment. So that again, in the families, in with children also. So in palliative settings, we give the best comfort possible to the child, so that the child in the last days remains comfortable without the toxicity of a hospital and needles and tubes everywhere and all those of things, which is completely avoidable if we know that, as per current evidence, if we go for intensive treatment also, there is no outcome. So these are two different streams of treatment that we have, and both streams has its own challenges. Many a times families may not be willing to accept that this is a palliated child. They may go around from hospital to hospital. In India especially, there is no referral network as such. So anybody is free to walk into any hospital and take treatment. So that in that kind of a scenario, whenever there is a rigid network system available, so then the patient can only go through that system. So and the system pays for the treatment majority of times. Even if it does not pay for it, there is a structured way of referrals. So if a doctor has said, the primary care person has said that okay, this is something has to be palliated, then it has to be palliated. They can get an opinion, maybe 2 or 3 opinion, but then it goes through the system only. But in many other countries, that's not the case. And we sadly see patients often who have to be palliated, but they are receiving high-end treatment, just adding to the emotional toxicity, the physical pain, as well as the financial toxicity. So these are challenges for that stream of treatment as well. Thank you.
Thank you, Dr. Nita, for throwing light on it. And Dr. Sushma, I think your personalized medicine treatment from the industry perspective can add a lot of solutions for the problems, the challenges in the oncology sector Dr. Nita was describing, right? Can you shortly describe that, and then we can go in for the Q&A session? Yeah, I think you're muted, Sushma.
Yeah. So what we can actually do is what we are presently doing is we have a pharmacogenomic test, as I was explaining in the introductory part. So this is a test which looks at the person's germline DNA, basically a person's individual capacity to metabolize drugs. So I would like to throw light on most of the important drugs which are being used in the clinical practice for cancer care. Anything which is a 5-fluorouracil, and this entirely depends on the metabolic capacity of a gene called DPYD. And this is responsible. So there are a lot of alleles which have been recorded by systemic groups, and these have been systemically used by us, and we look into all these variants to find out if that patient will be suitable to get this particular treatment, or what will be the toxicity profile. So basically, this would enhance the vision of a doctor to basically put them on the right dosage. One is right dosage means also you are correcting the disease at the point of treatment so that they're not getting a very lower dose. Because if they are a normal metabolizer, they can be treated with the right doses. They can also be dosed up. We have cases like that. And sometimes you need to know if the person is a poor metabolizer, where there'll be in toxicities, higher toxicity, they'll not be able to even tolerate, very low doses. And this can help in doing this right away without having any trial and error method. So basically, these tests are incorporated as initial screening mechanisms before the doctor could also prescribe a particular medication. And there are also stances where the targeted therapies, where, when we look at different molecular profiles, there was a particular patient who came to us. She was a patient, she had ovarian cancer, and she was being treated for first, second, third line. And this was a completely resistant, cancer. It was not responsive to most of the chemotherapies. And since we do a whole exome sequencing, we had the opportunity to completely sequence all the 19,000 genes, yes, and also look into the complete profile of the tumor mutation burden. So what we actually found is we found a mutation in the gene called POL. So basically, POL gene is found to be mutated in most of the colorectal and endometrial cases. But whereas here, we found this ovarian cancer sample had a hypermutation profile, where we found a POL mutation which was exactly in the exonuclease domain. So this domain is responsible for the functional part of the POL exonuclease. The complete function. So this was a specific mutation, which was a novel mutation also. It was not recorded, but then we were able to correlate, we were able to give the study, and this particular patient presented with a high TMB, which we could say it was more than 100 megabases per, usually it ranges from say like 10 is the lowest, and sometimes patients have 1, 2. And this also has a microstable, it was an MSS type. So with this, and we have recommendations from FDA where we can treat solid tumors which have very high TMB and they are eligible for immunotherapy. So this was such a case, and due to we had several discussions with the doctor to kind of understand why this happened, and we were able to see what was a particular gene that was mutated could have caused this very high hypermutated state, and the patient could be put on a therapy where she could be actually responsive. And this is also a type which is supposed to be a very chemo-resistant, subtype of ovarian cancer. In endometrial also, it's a very chemo resistant cancer. So these are the spaces that we have been dealing with. Yes, when there are approved medications and there are insightful discussions and papers which come up, we are able to give these discussions as clinical studies. We are able to send this across to the oncologist too, for them to take a decision.
Yeah, thanks, Sushma, for a detailed explanation. Now we can move on to Q&A session. We have some questions from the audience for the panelists. And Dr. Sushma, there is a question from the audience. What will be the cost of the test?
So the cost of the test, it ranges. Basically what happens is we do a whole exome profiling. It means that basically this could range from anything between, because it is a therapy for, which this is something which we give it for immunotherapy. And the immunotherapy costs for every session will be almost 1.5 lakhs. So a patient would want to know whether they are eligible for that therapy and if they would be responsive. So the tests, we have high-end tests, and we also have tests which are pharmacogenomic tests, which are also in different ranges. So we cater these tests in different ranges by what is the requirement of the physician, and or also what the patient is able to take up, because here we don't have a system to where you have the payers are the patients here. So depending on what is the exact thing that the doctor requires, and also he will look at what is the patient's feasibility. So it ranges from say like 20 to even like 1.5. So there's a different range of tests which you can take. It can be a comprehensive gene test for 10, 11 or 12, or you can look into the whole exome, or you can look into the 200 plus different medications for pharmacogenetic. So it ranges from a minimum price of 18 to 20, and then it goes high up to 1.5.
Yeah, thanks, Dr. Sushma. Thanks for throwing light on it. Next we have a question. There's a doctor from UK. They have given a feedback about the valuable insight she gained through the session. Thank you. And the question for the panelists: what role does research play with more standardized cancer care in Chennai, Tamil Nadu, and India? We have nice National Institute for Health and Care Excellence guidelines that we follow when treating patients. Like this is what they follow in UK. What is the kind of things that we have in India?
So basically, if I could actually take up this question, so basically guidelines, so these are built by global communities. So whatever we are using in India is something which is being used in the global community. What are the things? And we also have our own regulatory bodies which actually sanction most of these, studies. So there are a lot of studies which is related to looking at what is the genomic landscape which is in our country. So basically looking into studies where we can also include a lot of Indian population. So there is a gap between having the Indian genomic studies at the forefront, which where we are actually there are more of the Western population results which are still there. So there is some a gap that needs to be filled, and there are a lot of institutions which are now coming in to fill this gap, and we have a lot of institutions which have now sequenced at least 1,000 healthy genomes of from the Indian scenario. So these are some efforts which have been done to include our patient cohort and to also see what are the difference in ethnicities, basically, if it is from the Indian scenario. So as of now, what we actually follow is all on global scale, what has been recommended on a global scenario.
I can add to that.
Thank you, Sushma. Thank you. And
Can
this is a question for Dr. Vaishnavi. Are you aware of Oncovaria F test, which can detect 4 G types of women cancer very early, even stage 1, breast, ovarian, cervical, and endometrial from blood sample?
Dr. Neeta, did
Can
you have
you-
anything to add to the previous question? I know you had something to say.
Yeah, I was typing the answer then. So yeah, there is from a clinical perspective, there are a lot of Indian evidence-based research as well. So like NICE guidelines in the UK, there are Indian guidelines as well. So it depends on the disease. For example, there is I'm not sure if you have heard about it. There's something called National Cancer Grid, which is a grid of more than 100 hospitals in the country, which has initiated as in it's been spearheaded by Tata Mumbai, and it actually brings in cancer treatment centers from across the country together. And National Cancer Grid has its own guidelines for management of multiple malignancies. So almost all so I look after only pediatrics, but almost all other malignancies, solid tumors as well as leukemia, lymphomas, there are guidelines which have come out from National Cancer Grid. In addition, there is Indian Council of Medical Research that also brings out risk adapted guidelines for the country. So that's again there. And finally, for different subsets, like for example, I look after pediatrics, so pediatric hematology oncology is our chapter, which also has its own guidelines for some of the malignancies. So we have our own Indian guidelines as well. Then under SIOP, which is an international group of pediatric oncology, there is risk adapted treatment for developing countries. So many of our countries would fall under that. Under the purview of St. Jude Global, which is a US based organization called St. Jude's Children's Hospital, which has a global alliance with multiple countries and multiple centers in multiple countries. So there's something called ARIA, which brings out risk adapted guidelines again for the different regions. So there are there is local treatment also, which is there, which is also go with this. And so we don't really follow protocols which are there. So just because cancer, but today in UK, NICE guideline says that this has to be done, it does not mean that the child has to be given that treatment here. So the treatment which we follow here would be out of our local expertise and the biology of the disease that we are treating, which may be very different from what a Caucasian patient is, a British patient is. So that would be different. So all of this is adapted to the local issues, the local biology that we are seeing, and the expertise that is there with the team in the hospital in India. Thank you.
Dr. Neeta, I would just I would like to have a small interaction with you. Yes. So when we're talking about the Indian scenario, yes, we do understand that clinical practices do have this. But what about biomarker or targeted therapy, like, responses, adverse events? How much of this is being included in all these guidelines, and how do you actually take help from the biomarker or the perspective of how we include all these toxicity profiles based on a personalized medication care?
Yeah. Yeah. So I replied because for the, I think the NICE guideline is basically treatment guidelines, right? So that's why I told that. From biomarker or for what you have been doing, as in you're looking at targets, you're looking at whole exome sequences. So that's something which we again do very commonly for our patients, to look at what can be targeted for our patients other than the routine chemotherapy that we offer for our patients. What else can be given which actually reduces, which actually focuses the treatment in such a way that you don't have to give the entire gamut of treatment. You can just get away by giving a targeted treatment which would offer a better cure rate. So that again, there is local expertise that is building, right? So over the years, we have also realized which group of patients should be sent for the biggest challenge in targeted therapy currently is that for the newer ones, at least there are no generics, so it's extremely costly. But for the ones which have come earlier, for example, tyrosine kinase inhibitors came so many decades ago. So now we have so many good generic options in our country also which are available. So that is not an issue at all. And I think regarding your question on biomarkers and toxicity, yes, there is a lot of work. And like I said, toxicity, there is definitely a lot of work. And that is why there are treatment adaptations that are coming in. So you don't really need to treat the entire the complete treatment that has been given. We may not require to give, or maybe in some cases we may give more or less. So that adaptation happens with expertise. So now, and sometimes, people ask you, what is the best protocol to treat this disease? And the wise answer for that question is that the best protocol is the one which you are most familiar with. So we have some protocols where we have been following it for 10 years or 15 years. We know exactly in what step what is going to happen. So when they've done it so many times over and over again, the treating team almost knows what is going to happen. And that actually helps you predict toxicity and be prepared for toxicity when it happens. So that is something which is which again comes with expertise and experience over years, and the teamwork definitely, and that helps us adapt treatment strategies for our region better. And regarding biomarkers, there is work. There are biomarkers which predict, as in, for example, nutrition is a big issue. So nutritional, especially for the center that I work in now, we cater to extremely, as an 80% malnutrition is what we are looking at. So when we have faced with 80% malnutrition, there's no choice but to adapt your treatment in such a way that, or be prepared to handle malnutrition. And nutrition is something which is extremely important to go through this treatment, right? So that has to be taken as an important part of treatment. And so in our regular discussions, daily discussions, our nutritionist sits along with us to decide what we have to give for the day, the treatment. So that is how important it is. And there are biomarkers which actually can tell you what is which way the child will go. So that is also incorporated into our regular practice.
Thank you, doctor. Yeah, thank you.
There are a few more questions, but I think like we are held up with time actually. Monica, can I go with one question at least? Like, is it possible?
Sure. And also unanswered questions will be answered in our feedback email.
Okay. Yeah. Dr. Vaishnavi, are you aware of OncoVerix F test, which can detect 4G types of women cancer very early, even stage 1 breast, ovarian, cervical, and endometrial from blood sample? Company name Predomics has put on this question.
I just responded to the question. I'm not familiar with the test, and if there is any published data, I would like to know more about it. And in general, for the treatment guidelines, we do have a panel discussion that happens every year, and we do discuss any literature that's out there that has been published in the past 1 year, and any solid phase 2, phase 3 data that's available, any FDA approval. That's when we would incorporate all of that as submissions, and we would have a discussion with the entire team, and anything that needs to be incorporated in the treatment guidelines, that needs to be voted on. So we do have a panel voting that takes place. So it's a long process to include them in the guidelines. But no, I haven't heard about this test.
Okay. Vaishnavi, there is one more question on patient-derived on organoids. Like you were actually working on in vivo models, right? So can you quickly go through some 2 or 3 lines about like how these organoids are important for diagnostic and therapeutic decision making?
Yeah, so for the patient-derived organoids, the way how we would establish those ex vivo models is we do get tumor specimens. That's one way we can collaborate with the different clinics, right? So we do have, at least in my previous work, we had collaboration with different clinics and hospitals where they would send us the tumor samples, and we would grow them in a tumor-specific media in suspension, so in either in suspension or in a gel, in a matrigel format, where they can actually form these organoids in suspension or embedded in these gels. The media that we use is specific to each tumor, which has the tumor-specific growth factors. And once that is established, that's when we would have this organoid culture. And any kind of drug testing, we just like doing it in vitro setting, just by treating cells. Instead of treating cells, we would treat these organoids, which now it's a 3D structure, which sometimes you can also include other cell types like fibroblasts and other cells from the tumor microenvironment. So you're creating an environment which is very reflective of what's happening within the body. So that is the therapeutic aspect of it. That's where we would do preclinical testing. And the vision for this entire organoid testing is that eventually it becomes a diagnostic space where the aggressive vision for it is when there is a patient, you send the tumors for sampling. By that end, you would quickly develop those organoids, have a panel of drugs that you can test on these organoids, see which drug responds to these tumors, which can eventually go back to the patient. That's the therapeutic aspect that everyone's aspiration is on who are working in this organoid space.
Yeah. Thanks, Vaishnavi. Thank you. And we'll wind up with the section, I think. Yeah. And let me thank the panelists, esteemed speakers. Like, I think they're all very versatile experience and very in-depth experience. Whatever they have in their own domains, they have shared it. And it's a wonderful to have strong speakers like you. And from the diverse background, you contributed a lot, your knowledge and expertise. And I also thank the audience, like who have come out with wonderful questions, and definitely you have actively participated in the webinar. And I just want to tell the audience, like there are a lot of upcoming webinars conducted by GPR coming up, and stay tuned for our announcements. And the feedback form has been put in the Q&A box. Please do give your feedbacks. And there are links to GBR and iBlot social media pages. Please do go and access those pages. And thank you so much for all your presence and support. Yeah.
Thank you all. Thank you, Suganya, for very beautifully coordinating this and very thoughtful questions. So thank you for pulling in all of us, and we had a great discussion. It's very good to know about Vaishnavi and Dr. Neeta's answers, which were extremely helpful.
Likewise. Thank you, Suganya, and thank you, panelists. It was a great discussion. I learned a lot from today's discussion.
Thank you. Bye-bye.
Thanks, everyone, to the audience, the panel, and iPlot and Suganya. And have a nice evening or afternoon.
Yes.
Thanks for joining today.